Namaste to all students. Khushro Bitiya, myself Jayoti and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about gynogenesis, and this session is related to Plant Biotechnology course or program MSc BT second sem. So before starting gynogenesis process, you have to know about basic concepts of gynogenesis. So as we know, the dominant life form of higher plants is the free living sporophytes. The sporophyte is the resultant of fertilization of male and female gametes and contain a set of chromosomes from each parent, which genomic constitution is 2n. Cells of gametophytes carry half the sporophytic set of the chromosome, means they are haploid in nature n, and in diploid plants which contain two sets, means 2n, and of the chromosomes. On the other hand, haploid plants are defined as sporophytes having only a single set of chromosome, means these plants will have n number of chromosomes. So that's why we are calling them haploid plants, means gametophytic number of the chromosomes equal to the gametophytic number of the chromosomes. There are several strategies and methods which have been worked for the production of haploid plants. We have already known that there are two methods for haploid plant production, which is androgenesis and second one is gynogenesis. In our previous session, we have already discussed about androgenesis process. And again, I'm just giving a brief information about the androgenesis and gynogenesis. So in androgenesis, haploid production of plants through anther or microspore, means pollen culture, has been referred to as androgenesis. When on the other hand, gynogenesis method is basically haploid plant production from ovary or ovule, means from the female reproduction organ culture, have been referred to as gynogenesis. Gynogenesis is the formation of a sporophyte from the female gametophyte on artificial medium. It is a process by which culturing of unfertilized ovaries to obtain haploid plants, and it was first reported in barley by San Neom in 1976, but later works done in wheat, rice, maize, tobacco, etc. In most cases, the optimum range of ovary culture is the nearly mature embryo sac, but in rice ovaries at free nuclear embryo sac stage are the most responsive for haploid plant production. There are two methods for in vitro production of gynogenic haploids. They are ovary culture and second one is ovule culture. So before starting ovary and ovule culture process, you have to know about the difference in between term of ovule and ovary. So for understanding the difference in between these two terms, you have to know about the complete structure of flower and its reproductive parts. So ovary and ovule comes into female reproductive organs, where this complete structure is known as ovary, and this ovary is having these small kind of a beads structures which are known as ovule. In vitro gynogenesis is used as alternative technique in species where anther or pollen culture is unsuccessful. Means first of all you have to try anther and pollen culture, but when it is going to fail, so you have to move further for gynogenesis process. So for the haploid plant production, first of all just select the method anther or pollen culture. After that you can select the gynogenesis when these two methods are going to fail. And the gynogenic plants are may arise through direct embryogenesis, or the gametic cells may form a callus followed by plant regeneration. Means It can be direct embryogenesis or indirect embryogenesis. So both ovary slice culture and ovule culture can be carried out simultaneously for achieving in vitro gynogenesis process. There are two stages. First one is induction. So ovaries are located on a liquid medium, which is MS medium, Moraceous scoop medium, having low oxygen content and kept in dark. Second step is regeneration. So ovaries are transferred into agar medium with high oxygen content and kept in light. The experimental plant is kept in controlled condition for few days and a normal sized flower is taken for culture. The flower is surface sterilized with 5% sodium hypochlorite for 10 minutes and then washed with sterile distilled water 3 to 4 times. Using aseptic method, the pistil is kept on a petri dish after removing sepals, petals, and stamens. In the petri dish, only fully grown mature pistil should be taken. Then the excised ovary is inoculated on the basal medium aseptically. The culture is incubated at 25-degree temperature for 16 hours photo period with 2000 lux light intensity. If the ovary is unpollinated, it will not grow on a simple medium containing mineral salts and sucrose. With the addition of vitamin B to the medium, normal healthy fruits can be obtained. So in the process of gynogenesis, first of all, you have to select healthy parent plant, then check ovary or ovule development stage by histology method. Further, select appropriate flower bud and surface sterilize, which followed the inoculation of ovary or ovule in suitable medium. So basically, after inoculation, you have to go for incubation of ovary or ovule culture. During the incubation period, callus or embryo will be induced, and after getting these callus and embryo, further go for the regeneration process. After getting regeneration process, you will get haploid plants. There are some triggering factors which can enhance the results of or improve the results of gynogenesis process. So first factor is pre-treatment. So cold or heat shock treatments can be used as a triggering factors. Cold pre-treatment of the inflorescence or flower enhances the gynogenesis. For example, 24 to 20 hours at 4-degree temperature give the better result in sunflower, and 24 hours incubation period at 7-degree temperature gave better results in rice flour. Medium which can be used for gynogenesis process. So MS medium, N6 and B5 media are very much suitable for gynogenesis process. Sucrose concentration can be in range of 3 to 15%. You can use solid or liquid media, both are suitable. And if we are talking about the phytohormones, so auxin and cytokinin, and especially 2,4-D is very much good plant hormone for better results of gynogenesis. Here is a one note that auxin can replace the pollination stimulus growth regulators, which are known as GR, basically 0.125 to 0.5 mg per ml of MCPA, means 2-methyl-4-chlorophenoxyacetic acid, induce somatic calli and somatic embryoids. There are some developmental stages at inoculation can be a triggering factor, like late flowering, for example, sugar beet, rice, etc. Early flowering can help in mulberry and sunflower, and early maturing female gametophyte is selected for gynogenesis process. In vitro maturation of embryo sac. The development pattern of female gametophyte in excised ovaries in vitro has been studied in onion, rice, barley, etc. In an experiment with onion in which ovaries at different developmental stages were fixed at inoculation and then at 3-day intervals, in vitro maturation of the embryo sac was evident. At the day of 12, in culture, only mature embryo sac were present. The maturation process is same as that of in situ maturation and a higher frequency than in situ maturation you will get. Now come to the origin of embryo. So embryo sac causes haploid egg cell, but also other cells capable of forming a haploid embryo, such as synergids, antipodal cells, and non-fused polar nuclei. Egg cells are predominant source of haploid embryo, for example, beta vulgaris and allium cepa, and antipodal or synergids origin of embryos have been proposed in Hordeum vulgare or Oryza sativa. If we are talking about that, that embryos has been developed by synergids, so examples are rice, and if embryos are getting to originate from antipodal cells, so examples are allium cepa. Now come to the uses of gynogenesis. So the ability to produce haploid plant is the best benefit in genetics, and plant breeding, plant physiology and embryology studies can be done by this method. And study of the genetic recombination in higher plants can be done by this method, and haploids are used for mutation studies. So gynogenesis is a very important tool to go for this study, and heritability studies are simplified due to haploid plants having only one set of chromosome, hence recessive mutation are easily identified. If we are talking about the limitations, so successful in relatively small number of species, and frequency of responding ovules is quite low, approximately 1 to 5%. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it. Thank you.